Hello all of you and welcome to chapter 17. We're going to be talking about the evolution and diversity of animals. This is an amazing chapter. It's a very long chapter because we'll be looking at all these different groups of animals and categorizing them. The first thing is, what is an animal? So they're very diverse. I have some pictures here of animals. Do you think of an ant as an animal? Do you think of a skeleton shrimp as an animal? But they're very, very different, but they have a few things that are in common to absolutely all of them. And that's what we want to look at. Well, what are they? One is they have multicellular bodies. It's not a single cell organism to be an animal. It has many. It has multicellular bodies. Those cells that they have are eukaryotic. So if you remember, eukaryotic meant that they have a nucleus. And also, they lack a cell wall, which means they're not a plant, right? A plant would have a cell wall. Okay, many cells with a nucleus without a cell wall. And what else? And they have a blastula. Well, what is a blastula? This has to do with a stage of development. As this embryo is developing, what it looks like. So you really do have to go to embryology to be able to determine if this organism is an animal or not. So a blastula, think of a zygote, which is a fertilized egg. You have a little egg, it's been fertilized, and now it's going to start to divide, and become an organism. And in this case, this example, it's going to become a sea star. So you would have these developing cells, and then let's say you have 4 cells, and then it's going to develop into 8 cells, and then it's going to continue. Mitosis continues on, and it's going to turn into a blastula. And so there's that word again. I'm going to go to my pen. And this word blastula has to do with this embryonic developmental stage in which you have those rows of cells but hollow in the middle. Let's make an H for hollow in the middle here. Hollow, a hollow ball, and that is a blastula. Later on in the stage of development, it's going to not be hollow anymore because some of these cells are going to start moving inside to make the internal organs of that organism. And that's going to be called a gastrula. Okay, blastula and gastrula. And then it's going to continue to develop those internal organs, and it's going to develop into an adult sea star. But that is, I'm going to backtrack. All animals have a blastula. That stage of development has to exist. If not, it's not in that category. We know that animal life began in water, and there are many animal phyla that originated during the Paleozoic, which is 530 million years ago, in this time where there was lots of different organisms show up in the fossil record. So we're calling it the Cambrian Explosion because there seems to be so many different organisms that are emerging here. And in the timeline, we have it here at the approximate, 500 million years ago. Lots of different weird little organisms showing up. How do we classify them? That's what this chapter is about. Well, we classify them into 37 major phyla. Phyla is the plural of phylum. And these 9 are an example that we're going to look at more closely. We're going to look at everywhere from the sponges to the jellies to the worms, annelids, and all the way up to the chordates. Okay? 9 of them. And we looked at cladograms before when we did the fruit one, where you had to categorize the fruit depending on whether it had a thick skin or a thin skin, or whether it had little seeds or a center stone. And those categories are the difficult ones to come up with when you're trying to find the difference between these organisms. So let's look at these. We know that there's an ancestral protist. This is an ancient organism that becomes now multicellular. Remember, that's one thing that all of them have to have. And then we have these branches. So some of them don't even have a true tissue. And then the others all have true tissues. So that's a characteristic that we'll be looking at. A tissue, what's a tissue in your own body? Muscle tissue. It's a group of muscle cells performing a similar function. They all get together and they contract together. So they form a function and therefore they're forming a tissue. They work in unison. Well, some of these organisms don't really have a true tissue. They kind of each cell for itself, so to speak. And that is the example of the sponges. So we're going to look at, do they have tissues? Then we're going to look at the other node and look at symmetry, and then these other words here that we'll have to go over one at a time. So let's go to the next slides and look at that. As a definition, remember, a clade was a group of organisms consisting of one common ancestor and all its descendants. So we could have one clade here, for example. We could have another clade here, right? Or we could include all of these and call this whole thing a clade. Remember, because it includes a common ancestor and all of the ones that come after that. Or in this other case, common ancestor and all of them that come after that. Okay, the first branching point that we're looking at that I was telling you about is about tissues. And as I said, the only one without true tissues are the sponges. So all the cells that make up the sponges are sort of individual little cells living in a colony, and they sort of do their own thing, but they're not collaborating with each other in a true way to perform one particular function. The second branch that we'll look at has to do with symmetry. What kind of symmetry? Well, is this organism radially symmetrical, or is it bilaterally symmetrical? So bilateral means that it has two equal sides. So if you were to take a slice of yourself that goes all the way from your mouth, down your forehead, down your nose, to your mouth, to your chin, and down your sternum, and down your middle, you're going to have a right side and a left side that looks pretty much the same. That's bilateral. And that's different from what a jellyfish looks like. In which case, in a jelly, for example, there are many planes that you could slice it in this one, or in that plane, or in this plane. Do you notice? I'm going to try and draw that. So there's one plane, and then left and right looks the same. And here's a second plane, and left and right looks the same. And a third plane, you can keep doing that with however many planes you want, because it has a radial symmetry. You can't do that with an organism here like a lobster. You can't really slice it this way and expect right and left sides to be exactly the same if I were to take like its head off in a different plane. Okay, so this would be bilateral, and this would be radial symmetry. So that's going to be an important characteristic that distinguishes certain groups of animals. There is something I wanted to alert you to, which is one that's very confusing, and it has to do with the starfish. And there, it would seem like there's more than one plane like this, right, that you could slice it, making it radially symmetric. But it turns out that as a larva, as an immature, really it only has one plane that you can slice it. So it's bilateral, only one plane that would allow a left and right to look alike. So starfish, even though you might be tempted to call it, because you think of an adult, you might be tempted to call it radially symmetrical, it's actually bilateral. Okay, this clicker we usually do when we're in person class. Which of these has a radial symmetry? I'm going to eliminate C because this actually has, we consider it no symmetry at all. And then this would be, oops, This would be bilateral. This would be bilateral, and the radial symmetry would be the sea urchin. Okay, let's go to the third branching point, and this has to do with the embryonic development again, and it determines this third branching point. And we're trying to distinguish whether it's a deuterostome or a protostome. Okay? Deuterostome or protostome. And what does that mean? Remember, we have these cells that are developing, and then eventually they are going to indent, and some of them come inside to make the internal organs. Well, that leaves an opening here. When these cells sort of bend in here to form some of the interior organs, then it makes an opening here. Right? We can see it really well on this one too. So we want to know whether that opening, eventually, when this little organism turns into a full-fledged organism, is that opening the mouth? Is it that end of an opening? If it is a mouth, we're going to call it a protostome. Pro-interior. Protostome. However, in some of them, what happens is that this little opening here is not a mouth, right? The same one here is not a mouth. It's actually the anus. It's the other end. And so that is a deuterostome. Okay? So whether that opening that you see is mouth or anus in the gastrula. I do want to tell you that these layers, these colors that they drew in here, have to do with the development of the outside of your body, which is ectoderm, is going, all these cells are going to develop into your skin and your nervous system. In yellow, these that first come to the very inside of the body is endo, inside. And that's going to give origin to the digestive tract of this little organism. And then what fills in between here, in the more orange, is the mesoderm, or middle, and that's going to be muscles and reproductive system. Interesting how it does that. And it'll do it in all these different animals. So if you watch this development in a lab, you would see then that these for sure will develop into muscle. Amazing. The next division here considers whether, we're going to look here in the protostomes, whether the organism molts. I'm going to put an M for molt. Molts, oh gosh, it keeps moving on me. Molts or does not molt. Do you notice? So if you're an ecdysozoan, ecdysone is actually a molting hormone. So if you have ecdysone, you're an ecdysozoan, and the arthropods and the roundworms are molters. They're going to molt. And that was my picture here, molting insects. This is a cicada. Really weird how it has an exoskeleton. So it's very hard to grow if you are trapped inside your skeleton that's on your outside. So the only way you can grow is if you grow out of your skeleton and then grow a new one. And then grow a little bit bigger, and then grow a new skeleton, and then grow out of the skeleton and grow a new one, and you molt. So crabs molt, insects molt. This one's just coming out of its cuticle. So protostomes then branch into the ecdysozoa, and they molt. The other group is the lophotrochozoa, and they don't molt. Just think of it like that. There are some other characteristics that are important, but at this level, for this course, all we think of is not molting. So let's go back. Ecdysozoan molt, it's the insects, all the arthropods, the crustaceans, and roundworms. And the annelids, the mollusks, the flatworms are not molting. Okay? Now, what comes next? I'm going to cut this a video here, and what comes next is we're going to talk a little bit about each group. what are the sponges? What do they do that's cool? What are the cnidarians? What do they do that's cool? And kind of go up this road here, looking at these different organisms. So for now, thanks for listening, and move on to the next video. Thank you. Bye.